Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today, Install Africa. Install Africa, the Innovation Science and Technology for Action Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Install Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. Next, we will move on to narrating the profile of our invited guests.
Ladies and gentlemen, it is my honor to introduce a distinguished leader in global public health, Dr. Muhammed Afolabi, a physician, academic, and internationally recognized researcher whose work continues to shape child health policy across West Africa and beyond. Dr. Afolabi is an associate professor at the London School of Hygiene and Tropical Medicine. He earned his medical degree from the University of Ibadan, followed by a master's in public health from Obafemi Awolowo University and a PhD from LSHTM. He is a fellow of multiple prestigious institutions, including the West African College of Physicians, the UK Faculty of Public Health, and the UK Higher Education Academy. He currently leads a UKRI-funded project evaluating integrated approaches to controlling malaria and neglected tropical diseases in high-risk pediatric populations, advancing at a shift from vertical top-down disease control programs to sustainable, locally relevant strategies. Beyond research, he teaches and supervises at the postgraduate level and serves on high-level global health committees, including the WHO SAGE Working Group on COVID-19 vaccines, the UK Nuffield Council on Bioethics, and the adjudication panel for the pivotal R21 malaria vaccine trials in Africa. Please join me in welcoming a committed scholar, global health advocate, and changemaker, Dr. Muhammed Afolabi. The floor is yours.
Good morning, everybody. Please, can you confirm that you can hear me all right? And I want to say a big thank you to the organizer of this very important webinar series, the Install Africa and FIRAT Consortium. I'm highly impressed by the level of preparation and the technological touch that the organizer has put into this particular webinar series. So thank you very much, and well done for the excellent job that you are doing. So I'm here this morning to share with you my journey in vaccine research and I title the topic of my talk as From Bush, Bedside, Bench to Boardroom. From Bush means that we go into the field to collect data. And by virtue of my clinical background in medicine, I also treat the patient and the research participants at the bedside in the hospital wards. Not only that, I go into the lab and stay in the bench to perform laboratory experiments on the samples collected from these research participants. I do not stop there. The findings of the data that I collect from my participants in the bush, in the field, in the hospital wards, and in the laboratories, I ensure that these findings are translated into policies to ensure enduring public health impact in the boardrooms. So thank you for joining me in this discussion, and I'm looking forward to a robust and fruitful engagement. Thank you once again. So as a result of my presentation will be organized along this outline. I will share with you how it all started from my Gambia experience, where I led HIV vaccine trials in infants, following which I started the malaria vaccine trial in adult children and neonates. And from Gambia, I went to another place called Cambia in northwestern province of Sierra Leone, also in West Africa, to lead the Ebola vaccine trial in children and infants. And as God will have it, as I'm stopping from Ebola, COVID also started, and I was able to translate and leverage my knowledge and experience in Ebola control response in that of COVID-19, where I work in high-profile committees that involve on developing roadmap for COVID-19 vaccine supply in the context of a limited supply, as a member of the WHO Strategic Advisory Group of Experts, Working Group on COVID-19 Vaccine, and also as a member of the North-Field Council on Bioethics. So this is just summarizing what you have already known from the nice citation read. I started my education from the Premier University of Ibadan, where I obtained my medical degree in 1999, following which I completed the postgraduate medical training, they call it residency, and became a fellow of West African College of Physicians in 2006, and a fellow of National Postgraduate Medical College of Nigeria in 2007. During my postgraduate medical residency training at Obafemi Awolowo University Teaching Hospital, Ile-Ife, Nigeria, I also enrolled for a Master in Public Health at Obafemi Awolowo University, Ile-Ife, which is not far from each other, and I completed that degree in 2007. I took up a consultant role at Ladoke Akintola University in Osogbo, Southwest Nigeria, from 2007 to 2009. But out of curiosity, I took up full research appointments with the Medical Research Council unit in the Gambia, West Africa, from 2009 to 2016. And I'm going to talk more about it in subsequent slides. During my stint in the Gambia, I got a PhD fellowship award, which took me to London School of Hygiene and Tropical Medicine for my PhD between 2011 and 2015. A day after I defended my PhD thesis, I was offered a job at the London School of Hygiene and Tropical Medicine to lead an Ebola vaccine trial in Sierra Leone, because that time corresponded to the peak of Ebola outbreak. In fact, it is the largest Ebola outbreak in West Africa. It happened between 2014 and 2016. By January 2015, I was offered a job after my PhD thesis at the London School of Hygiene and Tropical Medicine. I did not take that job until 2016, and I was there as a staff of London School of Hygiene and Tropical Medicine, but based in Candia, Sierra Leone, from 2016 to 2020. So while in Candia, Sierra Leone, I developed a proposal that leveraged the delivery of deworming drugs for children that are co-infected by malaria and neglected tropical diseases, mainly soil-transmitted ailments as schistosomiasis. And that was submitted to the UKRI Future Leaders Fellowship Scheme, and that attracted an award of a fellowship of a grant value of about £1.4 million. So I relocated to London in January 2020. I barely spent about 2 months in London settling in when COVID spread to the UK and the whole world was shut down. So I've been in the UK since 2020, but I try to cascade this or chronicle the stages in my experience so as to provide the context and background to the discussion that we are going to have today. So the next slide is showing the work that I led as the clinical research lead on phase 1 HIV vaccine trial among infants that are not infected with HIV and whose mother were also not infected. As you know, phase 1 trial are usually the early phase trial after the preclinical testing have been done in animal models, either the macaque monkeys or non-human primates. So I was fortunate to be one of those who led this very important novel vaccine trial among children in Gambia. And as you can see on the screen, the HIV vaccine is called MVA-HIV-A. MVA stands for Modified Vaccinia Ankara. It is a vector, or what we can call a carrier, on which the HIV immunogen was coupled so as to deliver it into the infant. And when this vaccine is delivered, it will generate antibodies that can sort of protect the child from becoming infected with HIV when they are exposed to HIV virus later in life. The HIV immunogen is made up of HIV-1 GAGs and the CD8 T cell epitopes. I do not want to bother you too much, but the important takeaway message here is that the vaccine is MVA-HIV-A, and it's a recombinant MVA vaccine. It contains coding sequences of the immunogen HIV-A. The meaning of all those jargons that I've just mentioned is that this particular vaccine cannot cause HIV on its own because it was made in the lab. It's recombinant technology that was used to develop it. So it cannot cause HIV in any person that they are vaccinated with it, because it just looks like the HIV. It was not HIV on its own. So in the Gambia, we recruited a total of 65 mother-child pair, and we screened them and excluded 17 of them for various reasons. And we randomized 48 out of them into two groups, vaccine groups and no vaccine group. And we follow all of them for 365 days. So if you look at the diagram below here, it shows how we conducted the trial. At 16 weeks, they were given their routine childhood immunization called EPI in Africa. And at 19 weeks, we screened them, collected their blood sample to ensure that they are healthy in terms of the football count, the biochemistry, and also the EPI antibodies. And by 20 weeks, we now randomize them into two by chance, so that we are not biased, that this child is receiving the HIV vaccine and the other one is not receiving it. And we follow all of them, irrespective of whether they receive the vaccine or not, continuously until 365 days. But at some time point, we were collecting blood sample at 21 weeks, 28 weeks, 36 weeks, and follow them up until 365 days. That is 52 weeks. And the main reason of why we are following them is, one, to ensure that the vaccine that we gave them was safe, and also it elicits adequate immune responses. That's what we call immunogenicity in these children. Because we need to have this finding before we can move to the next phase, that's phase 2, whereby we'll be able to increase the number of a vaccine that we are going to give to these children. That is, we started with small number of 48, but we need to step the number, having satisfied that the vaccine is safe and immunogenic. So this is the result of what we found. We found that the vaccine MVHIV was safe and well tolerated, but it did not elicit adequate immune response. And the reason being that we only gave one dose of the vaccine. If we have done a kind of a technology called prime boost, that is, we first of all prime the immune system of the child with the first vaccine, and we give a booster dose maybe 4 or 8 weeks apart, it would have elicited more immune responses. So that's one of the learning points. But we are able to establish the safety and the tolerability of the vaccine. We were also able to establish that the HIV vaccine does not interfere with the antibody of the routine childhood immunization that we are giving concurrently with the HIV vaccine. So the lesson that we learned was that this particular vaccine needed to be given more than once. And it now provided a kind of a platform to move to the next stage, whereby we now use another kind of technology to replace the HIV-A immunogen and use a kind of the immunogen at a universal that can focus on all the kind of vaccine strain, I mean HIV strain, that we are not covered in HIV-A immunogen. And this we are called universal pan-clade immunogen, and that is currently where the vaccine, HIV vaccine effort is. On completion of that HIV vaccine trial, I was fortunate again to work on a malaria vaccine. You all know that malaria vaccine is very important and is one of the strategic tools that has been found to be very, very potent and effective in controlling the burden and the mortality due to malaria. And we know that Africa, especially sub-Saharan Africa, has the burden of malaria vaccine of up to 95% of the global burden and up to 94% of the global mortality due to malaria can be found in sub-Saharan Africa. And apart from all the other tools that have been used for malaria control and elimination, including the use of diagnosis tool, anti-ACT, artemisinin combination therapy, indoor residual spraying, as well as other vector control, we find out that vaccine seems to be the most potent. And for over 4 decades, scientists have been working on getting an effective vaccine that will be able to achieve up to 75% efficacy for the control of malaria burden and mortality. So the precursor of R21 that we currently have as the second licensed vaccine after RTSS is what I'm going to describe. And it consists of 2 main vaccines. Recall that I mentioned that MVA, HIV-A is a single vaccine. But this time around, we have a prime boost vaccine called chimpanzee adeno63 ME trap and MVA ME trap. So, and I started that, you can see the chimpanzee adeno63 ME trap is the one that has all this strength, like that of Ebola virus. And it's what we give as the first vaccine to prime the immune system. And 8 weeks after, we give the MVA ME trap, the same immunogen, but with different carriers or different vectors. This type of technology ensures that the same immunogen is delivered 8 weeks apart by different vector, and this helps to ensure that the body induces adequate protective antibody or immune responses that can challenge malaria parasites. And by the way, this type of vaccine is called the liver stage vaccine or the pre-erythrocytic vaccine. That is the vaccine that works before it gets into the red blood cell. Immediately the malaria parasite gets into the blood, it goes into the liver. So, and that is where this vaccine works. So in the Gambia and in Kenya, we did the first one for the vaccine in adults. And this is the, you can see up is the Gambia trial flu chart and Kenya trial flu chart. Okay, I'm receiving information here that the slide is not moving on the YouTube, but I'm the one explaining here. I've not yet moved it. So I'm going to move it now. Can you confirm whether it is moving or not? Is it moving now? I've moved to the next slide. Yes, thank you very much for confirming. Okay, this is now showing the results, the safety findings after we have given this vaccine, malaria vaccine, to adults in Gambia and in Kenya. We find out that the vaccine elicits very, very low safety concern by showing that it only causes mild swelling and mild pain. And also when it is given the second time, it's also causing mild symptoms, fever and pain at the injection site, and this resolves within 24 to 48 hours. And this is just like any other injection that we take. So we don't want it to be something that will be given to somebody and it will now cause them to develop a kind of serious and severe safety concern, like anaphylactic reaction, that person will develop goose pimples and start having respiratory difficulty and may die eventually. But we were able to establish that the malaria vaccine given in 2 different populations, in West Africa, in Gambia, and in East Africa, in Kenya, shows that there are no safety concerns. And when we now increase the dose of the second component of the vaccine, the second dose of the vaccine, the booster dose, we find out that the frequency of this safety concern, they just increase minimally, but not very severe. So that is an important and a promising findings that was also supported by the result of early spot responses, showing that there's substantial immune responses that is seen in adults. And there are no statistical significance between the peak immune responses in the individual who receive the vaccine, either intradermally or intramuscularly. The immune responses in African adults was also compared favorably with the malaria vaccine exposed or malaria exposed adults in the UK. And on the basis of this, we move to the next age population, that is the children, because we don't want to start with the children and we don't want to do it only in adults, because if you do it in adults, the physiology of the immune system in adults is different from that of children. So that's why we use the age de-escalated design. This implies that we started from adults, we moved to the children, and the children are divided into various groups. There are adolescents, then we move to the young children, and we move to the infants and to the neonates, which are the newborns. And we find out that malaria is very, very prevalent and cause more problems among children who are less than 5-year-old. And because these are the target population, we do not want to start from those very vulnerable age group. We started from the adults and we move gradually after confirming that the malaria vaccine is safe in the older age group. We move to the next age group, and that was how we conducted the trial. So the next one was also in Gambia and in Burkina Faso. And in Gambia, we recruited 140 children in different age group. One, age group 2 to 6 year old. Second age group, age 5 to 12 months old, and 10-week-old children. We divided them into the very different age group. And you can see it's a very, very complicated and complex trial that require a lot of meticulous planning in terms of the recruitment. And when we start with the first age group of 2 to 6 year old, we will give them the vaccine, and we wait until 8 weeks after we give them the second dose of the vaccine. And if this vaccine show demonstrable safety, and we will now consult a group of experts called Data and Safety Monitoring Group. They will look at all our data and they will now give us the go-ahead to move to the next age group. And that was how we started with age 2 to 6 year old. We moved to age 5 to 12 months old. After confirming that it is safe in the first age group, we moved to the next age group. After confirming that it was safe in the age 5 to 12 months old, we moved to age 10-week-old. That was what I did in the Gambia. As I was doing that in Gambia, I have colleagues in Burkina Faso, specifically in a place called Banfora, which I've traveled to many times. They were also doing the same thing. And at the end of the day, we now compare the result that we found in the Gambia and in Burkina Faso. So this is the result. And the result shows that the immunogenicity responses or the immune responses for those in the 2-year age group and those in the 5 to 12-month-old group and 10-week-old age group, as well as 5 to 17-month-old Burkina Faso, it showed that it demonstrates that when the vaccine is given after the 56 days, it goes to a maximum point. You can see in all of these charts that when you give the first vaccine, dose of the vaccine, the immune responses rise up by 28 days and it falls slowly. And by the time you give it the booster dose, it goes up again, and the maximum period corresponds to day 63 before it starts to wane, and it can continue like that until 365 days. That is what we were able to demonstrate. And that shows that, one, the vaccine that we are going to have must not be the vaccine that you give only once, because we want to ensure the durability of the antibody responses. And when you give the second dose, it reaches a maximal level or a peak before it wanes. And we follow up these children for up to 365 days to see how well the vaccine is waning. So after 365 days, when the vaccine wanes so low, we also recommended the giving of the third and the fourth doses of the vaccine. All these studies provided the platform for the current vaccine that we are using, and that was why we were giving them in doses of 4. Because if you get only 1 dose, it will not give adequate protective antibodies against malaria parasites. So the last one I'm going to share with you is that of the malaria vaccine in newborns. One might be wondering that why are we so behaving like a crazy set of people, that a newborn, how can a newborn have malaria? But studies have shown that malaria can be transmitted from pregnant women, a pregnant woman, to their babies while the child is still in the womb through the placenta. And we have established that a child that is born today can have what is called congenital malaria. And if they are not treated promptly and adequately, that child can die before the 7th day of life. And that was why we now see that, having established the safety and the immunogenicity of the malaria vaccine in children that are older than 5-year-old, that is the adolescent, in adults, in also children that are 2 to 6-year-old, 5 to 12-month-old, and in 10-week-old. We also want to see how this malaria vaccine will fare when we give it to children who are born today. And that's what we did. So we follow up pregnant women who comes to our facility from the age of gestational age of 32 weeks. And as soon as they deliver, we will screen the children and we ask them to come when the child is 7-day old, and we give these vaccines to them. You can see how complex the step that we took in implementing the trial. We found very interesting results that is also similar to what we obtained in the other age group, especially the children that are 2 to 6-year-old, 5 to 17-month-old, and the infant, as you can see on this slide. So the summary of everything that I've been saying shows that the malaria vaccine of a prime boost heterologous involving chimpanzee adeno63 and the modified vaccinia ankara expressing the ME trap shows good safety profile when they were tested in adults children, infants, and neonates living in sub-Saharan African countries, not only in Gambia, but also in Burkina Faso and in Kenya, where we conducted the study. We also demonstrated that the vaccine, malaria vaccine, elicits strong immune responses across the all age group. And very surprisingly, the children who are 10-week-old expressed or demonstrated the peak of immune responses at day 63 after the second dose of the vaccine is given. And there's an important point that I want to make here, that this work that I'm presenting was the one that led to the second dose vaccine, second generation vaccine that was licensed by WHO called R21 matrix M. It was developed by the same group, Oxford Vaccine Group, led by Professor Adrian Hill, and it achieved 75% vaccine efficacy and has been approved by WHO as the second licensed malaria vaccine since 2022, and is currently being deployed across many countries in Africa. This malaria vaccine was also used to develop vaccine for Ebola, which I'm going to talk to you shortly, and also the Oxford AstraZeneca COVID vaccine. The same principle, the same platform were used in all the three vaccines: malaria, Ebola, and COVID. So moving on quickly, I will move to the Ebola. I conducted this vaccine trial between 2009 and 2016. As I mentioned during the introduction, I did my PhD concurrently while I was conducting the malaria vaccine trial in the Gambia. I'll come to London for my stay in London and go to Gambia to collect my data. And I defended my PhD January 8, 2015. And a day after my thesis defense, I was offered to lead an Ebola vaccine trial in Sierra Leone, because then Ebola was ravaging 3 West African countries of Sierra Leone, Guinea, and Liberia. And London School was contacted by Johnson & Johnson Pharmaceutical Group to conduct this Ebola vaccine trial that has similar, a lot of similarity with the malaria vaccine I conducted. So briefly, I want to tell you about Ebola disease, which you are likely to know. But since 2022, 2023, there has been a kind of a change in the naming or nomenclature of Ebola disease. Before, we used to call all the Ebola disease Ebola virus disease. But now there has been a change in the nomenclature. Ebola disease is now described as a group of deadly diseases. The fatality or the case fatality rate could be between 25 to 90%. That is, if somebody is infected with Ebola, 9 out of 10 of those people will die. And it is caused by a virus, an RNA virus, within the genus of Ebola virus. There are 6 main of member of this family. And the first one is the Zaire Ebola virus that was recognized, or that kind of discovered in 1976. I will discuss that shortly in my next slide. And the one that is called by Zaire Ebola virus is called Ebola virus diseases, unlike the name that was given to all of them before. The one that has been happening since 2022, 2023 in Uganda is caused by Sudan Ebola virus, and that is not called Ebola virus disease, it is called Sudan Ebola virus or Sudan virus disease. There's another one caused by Thai forest virus, and that one is called Thai forest virus disease. Another one called Budimbuyo, this virus is called Budimbuyo virus diseases. The other type are resting virus and Bombali virus. Ebola, Sudan, and Thai virus, as well as a Budimbuyo, cause disease in human being and non-human primates, like monkeys and chimpanzees. But resting virus does not cause disease in human being. It causes disease in non-human primates and pigs, but there have not been any reported cases in human being. Bombali virus, Bombali is the name of a village in Sierra Leone. And Bombali virus was detected when I was in Sierra Leone in 2018 in some bats. And it has not been known to cause disease in either animal or human being. So please take note of this description of Ebola diseases and the change in nomenclature. So the next slide is going to talk briefly about the history of Ebola disease outbreak. As I mentioned, it was first described in 1976 near a river called Ebola. That river has dark water, and that was why they name it Ebola. And disease is actually rare, but there have been a series up to 32 outbreaks, excluding the one that was reported in January 2025 in Uganda in a male nurse in a place called Mulango in Uganda. The nurse eventually died, and there were 43 contacts. The largest outbreak in West Africa occurred, as I mentioned earlier, from 2014 to 2016, and it reportedly confirmed cases of up to 29,000 people were infected, and more than 11,000 deaths were reported. There have been a kind of a trend in underlying drivers of Ebola, which included a kind of disruption of the ecosystem between human being and that of animal. Because of this change in human behavior, there have been a kind of increase in the risk of Ebola from up to 1.75 to 3.2 increase in food. That is, before, about 2 times risk is what we have of having Ebola outbreak. But now, because human beings will always go to hunt animals that are likely to be infected, the risk has increased to more than 3 times. And this is likely to be higher by 2070, especially in Africa. So in 2016, I joined the Ebola vaccine team in Sierra Leone to work on this Ebola vaccine trial that was developed by Janssen or Johnson & Johnson Pharmaceutical Group. And as you can see, it is also similar to the malaria vaccine that I shared with you because it has 2 doses. The first dose consists of adenovirus 26 as its vector. The malaria vaccine, if you recollect, consists of adeno 63, and this one consists of adeno 26. The immunogen consists of Zaire Ebola virus, while the second dose consists of MVA modified Vaccinia ankara. BN is a Bavarian Nordic phyllo. And these vaccines are given 8 weeks apart, with first dose consisting of R26 Zibov, and the second dose consisting of MVA BN Vilo. And we did this trial in a place called Cambia. And the selection of that place was based on the mathematical modeling of the trends and projection in the Ebola transmission at that time. And we were able to use that to find that the hotspot of Ebola transmission was actually at the border of Sierra Leone and Guinea. And that place is called Cambia. Cambia is a place that is about 10 minutes from the border of Guinea. And that was one of the places where the people of the Guinea came through to Sierra Leone, and that was how the virus transmitted or get into Sierra Leone. So this is also showing some of the initial work that we did to do the trial design. And not to bother you too much, we did the trial in 2 stages. The first stage, we started with adults, just like in the malaria. After we find out that the vaccine was safe and elicits adequate immune responses in the small number of adults, we now move to a large number of adults, about 400, and 576 children, and that's the component of the trial that I led. And these were broken into adolescents 1 to 17 and children 12 to 17, 4 to 11-year-old, and 1 to 3-year-old. And we were able to establish, just like that of malaria vaccine, that this Ebola vaccine is safe because the safety symptoms they were having were mild to moderate, and there were no serious or severe safety concern. And on the basis of that, we also find that it elicits adequate immune responses. As you can see, that when the first vaccine dose is given, the immune response will shoot up. That is in the blue color. The red color is the one that is given as a kind of a placebo. That is the meningitis vaccine to compare. And that one falls down. And when it is given as the second dose, it shoots up until about 365 days, when the vaccine antibody wanes gradually. And in adults, we demonstrated that at 21 days and 12 months post-dose 1, the antibody responses were observed in this number of adults. And that's made us to move to the next group of participants, which is the children. And we now find out that it also elicited the same level of immune responses across the age group. You will see in green is that of 12 to 17. In blue is 4 to 11-year-old. And in red, representing 1 to 3-year-old. We find out that at 12 months after giving the first dose, the responses were sustained. The antibody responses, the protective antibody responses were maintained in up to 70 to 96 of adolescent children and the younger children called toddlers. And the antibody, neutralizing antibody also reflected the same thing that I've mentioned. We published this paper in Lancet, and it was widely accepted. It attracted international attention, and it led to the authorization of the vaccine by European Medicine Agency in July 2020 and WHO pre-qualification in April 2021. And this vaccine has been used in various Ebola outbreaks in DRC Congo. When there was also outbreak in Uganda, the government of Rwanda also got this vaccine, and they use it to protect their citizens. And that's why you will see that unlike 2014 to 2016, that we have the largest outbreak of Ebola in West Africa, we have not seen similar outbreak that has gone out of proportion. This is showing that vaccine is an important and potent tool for the control of infectious diseases like Ebola. So moving on to the next stage, we did some other sub-studies. I don't want to waste too much of your time. I will just summarize it, that among the people that do not have Ebola, we find out that they were having Ebola antibodies, and we did not give them Ebola vaccine. How did this happen? We find out that some Africans, they already have natural Ebola antibody in their system without being exposed to Ebola or without being vaccinated with Ebola vaccine. And this also shows that if we want to give Ebola, we need to put this into consideration. And we also find later that if you are giving Ebola, the Ebola antibody increase with age. And this association is not observed with whether you are a healthcare worker or whether you have had Ebola before or whether you have attended a funeral of somebody suspected of dying or from Ebola. However, we find that people who live in household who have animals like pigs, they tend to have some kind of a correlation. That's the finding that we were able to get from our study. As I mentioned to you, that on completion of the work in Sierra Leone, I got a fellowship that facilitated my relocation to London for a fellowship. But I've not settled in for more than 2 months when COVID struck. And during that period of confusion, and I was now reflecting on how I can use my Ebola response experience to leverage and use that to a kind of a COVID-19 pandemic response. And I authored this paper along with colleagues, where I identified some important lessons that we were able to demonstrate in Ebola response in Sierra Leone, including the massive public health response, facility and ambulatory care, what's to be done to implement clinical trial during disease outbreaks, rapid data sharing, global health security, building reciprocity for data and samples, and development of regulatory system within African region. I advocated along with my colleagues that helped us, and that could also be useful for COVID-19 response. I was pleasantly surprised about a week after to receive an email invitation from WHO to join the membership of a WHO Strategic Advisory Group of experts, working group on COVID-19 vaccine, representing Africa. Only 2 of us were of African descent. I also find out that about a year after, some of the recommendations of this publication were also implemented by WHO, which identify 6 African countries to begin the manufacturing of mRNA vaccine as a part of a WHO scheme. So the countries include Egypt, Kenya, Nigeria, Senegal, South Africa, and Tunisia. They were now global mRNA hub, not only for COVID-19 manufacture, but similar vaccine that can use mRNA platform. This is showing how findings of studies can translate into public health impact, can translate into policies that can have enduring public health impact. While I was at WHO working as a member of WHO working group of COVID vaccine, I work with colleagues across all the parts of the world to develop many technical guidelines and policies. And when I counted them, there were more than 40, some of which I listed here, including the guidelines on the usage of the vaccines that were developed by Janssen, by AstraZeneca, by other vaccine developers like Pfizer, BioNTech, Bharat, and so many other ones. Before any of these vaccines can be used, they need to come to WHO. They will present the findings, and they will also present the safety and the immunogenicity findings, which will be discussed extensively by the members before they can be approved for emergency use. And one of the challenges that I faced while I was on the WHO SAGE working group was lack of data from Africa. The two of us were persistently challenged to provide safety data on Ebola vaccine in Africa. And I contacted my colleagues across West Africa, but I found out that people were receiving the vaccine, but there were no documentation about the safety of the COVID vaccine. And that became very challenging because we do not have reliable and verifiable data from Africa. WHO has to be relying on data from other parts of the world to develop policies for use in Africa. And I took up the challenge, working with colleagues to now see that, okay, what is happening? Can we get some data, let's say across West Africa, the 15 West African countries, look at the uptake and the trajectory of the COVID-19 vaccination coverage, which were not available, which WHO needed to sort of plan for the next phase of COVID-19 distribution. And we developed this, and we find out that the vaccine coverage and uptake in the 15 West African countries was very low, and that each of the countries will have to increase their vaccination rate by 7 or 10 factor to achieve the target of vaccinating 60% of the population. And this target was needed to end the acute phase of COVID-19 pandemic then. And then in high-income countries like UK, US, Europe, the vaccination coverage was about 60% or more. But we find out that in Africa, especially West Africa, it ranges between 0.6 to about 1%, except in few countries like Cape Verde, where their vaccination coverage was very high. So as a follow-up to that, we extended the study to all the African countries. You know, I said we started from West Africa. We now look at how did COVID vaccine was implemented across 52 countries in Africa. And we find out that the vaccination coverage was also low. And it will take even greater number of increase in vaccination coverage to achieve the target. But in all this, we find out that Africa was not severely affected by COVID, unlike what was seen in UK, in US, and Europe. A lot of people add these factors to this. They said it was because African climate or temperature was very hot. But we have not been able to establish the reason for this. But what has been sort of considered as an assumption was that Africa most likely must have been exposed to some strain of coronaviruses. And there's a kind of a cross antibody protection to COVID-19. And that is why, despite the fact that there's a lot of COVID-19 transmission in Africa, it does not translate to a lot of severe disease in terms of those needing hospitalization, those needing oxygen, and those who die eventually from COVID-19 vaccine. That is the scientific assumption so far concerning the relatively low burden and mortality of COVID-19, despite the fact that there was low uptake and low coverage of COVID-19 vaccine in Africa. Moving on, I think I have 2 more slides before I finish. This is one of a prestigious committee that I also become fortunate to be a member. As you can see on the slide, I happen to be the only member of African descent. And Norfolk Council on Bioethics is an independent body that develop policy on kind of biological products, medical products that happen in UK and beyond. And one of the tasks that I was assigned to do is to develop the international perspective, how bioethic thinking and expertise inform research, policy making, and policy and public debate during COVID-19 pandemic. And I was able to establish that global vaccine equity had serious challenges in the face of a vaccine nationalism, because then a lot of high-income countries went and paid for vaccines that have not even been developed in the major vaccine among the major vaccine developer. They bought like 100 million doses of the COVID vaccine, and they were giving their residents. By the time UK was giving the second, the third, and fourth dose to children or to adults in the UK, people in Africa, they've not received first or second dose. And the people or the government of UK felt that they need to protect their residents, their citizens first. And that is what is called vaccine nationalism or vaccine appetite. And I made them to realize that if you protect only the residents of UK and the other people outside UK are not protected, and you are opening your border to allow people to come in, those people might bring in COVID virus, not vaccine, into the country. And the people you think that you are protected are not truly protected. And during that COVID-19 pandemic, it corresponded to the time of the first tenure of President Trump, when he withdrew social capital, support and influence and funding from WHO at the peak of pandemic. There was also lack of genuine international collaboration for surveillance, for data sharing, and sharing of life-saving public health measures like COVID vaccine, like PPEs, like face masks. Low and middle income countries were just left on their own to develop their own find their own way out. And there was poor pandemic preparedness, not only in Africa, but also in developed countries. We find out that developed countries like the UK, US, and Europe that prided themselves to have very strong pandemic preparedness do not actually have strong pandemic preparedness, as we see in high mortality that were recorded across this country. And I recommended that we need to continue to conduct rigorous, expedited research without losing the sight of scientific and ethical standards. And we should allow appropriate dissemination of research findings to navigate the kind of widespread of misconception, misinformation, and disinformation and malinformation that is called infodemic. So I would like to thank you once again for your attention and acknowledge the good people that I work, the partners, the funders, the colleagues from Ministry of Health in Sierra Leone, my colleagues at University of Sierra Leone College of Medicine and Allied Health Sciences, my colleagues at London School of Hygiene and Tropical Medicine, the vaccine developer Johnson & Johnson, the partners for the logistics, World Vision, Grammy Foundation, Goal, INSEM, University of Oxford, the funder are Innovative Medicine Initiative, and European Union Commission. Thank you very much, everybody, for your attention.
Sir, this is more than a presentation. It is quite very motivational, inspiring, and life-saving. As personally, I've learned a lot. I've got to understand some new terms that I never had the privilege of learning before, and that's part of the take-home from this presentation. On behalf of the entire team at Install Africa, I'm expressing our gratitude to you for the time taken to prepare this presentation, as well as the extent of the knowledge and experience that has been shared today. So my name is Taufik Tokoya Debwega. I'm a member of the Install Africa team, and I will give opportunity to reiterate a brief of what has been mentioned before going into the question and answer session. The title of the presentation is From Bush, the Bedside, Bench to Boardroom. My experience, my vaccine research story by our own very revered Associate Professor of Global Public Health at the London School of Hygiene and Tropical Medicine at the United Kingdom, Dr Muhammed Afolabi. It's your presentation and the title also reminds me of the popular sayings that it takes a village to raise a child. And I must say that Africa as a continent, and Nigeria in particular, is proud to have raised you, and you are of benefit to the global space. The research interests have been indicated majorly largely in child health, in ethics, immunization, vaccines, vaccinology, clinical trials, public health, epidemiology, implementation science, as well as the aspect of randomized control trials. We can see how your doctoral experiences was able to cascaded to greater opportunities, particularly when we had this the Ebola outbreak and subsequent challenges that faced the world. We are delighted to have you, and we hope that this is going to be the beginning of several other engagements to come from the Install Africa team. And I want to, on behalf of the entire team, say that a physical contact is necessary at this time to be able to have very closer interaction in learning more from the experiences that we have gathered over the years. That is of greater pride to the African continent. I must say that you are on a loan. You are on a loan to London School of Hygiene and Tropical Medicine, and you can see the effect that the WHO was able to easily pick. That no, let's transfer you back. And I marvel at the extent of the trajectory trial that was done, starting from the West African region, and now how this study were expanded to the entire region in Africa. So which still shows that several more are still needed to be done. And we hope that with your availability for mentorship, several will be able to learn more and then be able to translate research into what will guide policy effects across the continent. While that is on, I would like to read some of the questions that we've had listed on the chat box. First is a concern about how do we, what are the steps, the step by step approaches to vaccine development? That's the first question, sir. From Hamza Alaji Abdullah. Thank you, sir.
Thank you very much, my dear brother, Dr. Taufik Tokwe Adeboyega. Did I get it right? Thank
Very
you so
correct,
much.
sir.
Yes, sir. Yes, sir.
So thank you so much, and also want to really appreciate our dear sister, Dr. Kudirat Adedeji. So, and thank you for the invitation, and I really appreciate the excellent work that you are doing. I've gone
Thank you,
through
sir.
your website, and I'm really, really impressed. So, and what always amazes me is that Nigerians, we are doing extremely well outside the country. So how do we leverage this to our country? Nigeria need us, and not minding that the environment back home may not be conducive. We need to replicate and leverage as many of these expertise. So Dr. Kudrat Adedeji have demonstrated very high level of competence and professionalism since he contacted me, and I was really, really impressed. I've been having talks with private organization, and I can say that in South Africa, FIRAT, being led by Dr. Kudrat Adedeji, I'm not surprised because the spouse is also an excellent leader par excellence. You know, when they say excellent leader par excellence, so it's like runs in the DNA in the family. So I pray that God Almighty will continue to strengthen you, and that FIRAT install Africa will continue to grow from strength to strength. So please continue the good work you're doing. I'm happy to provide my support in my own
Thank
little
you
way.
very much, sir.
And just to respond to Dr. Adeboyega, that you can also knock on my door. Please, my door is open. You don't even need.
Thank you.
I operate open door policy, and I'm happy to sort of collaborate well with you. So, and going to the question. So, thank you very much, my brother Amsa Alaji Abdullah, about what are the step-by-step approaches to vaccine development. Thank you. I'm sorry that I did not start from there. I would have done that. So, in a recent talk that I gave at Embark, I think last year, June or thereabouts. So that's one of my first slide where I highlighted step-by-step approaches for vaccine development. And the first step is to identify the antigen, that is the active ingredient. You know, I was mentioning MVA, HIV-A, adeno 26 ZIBOV. That adeno 26 is not the active antigen, it's just a vector or a carrier. It's like a vehicle, like a car, or that will now deliver the parcel to where it is going. So that MVA is also a carrier, a vector. But the antigen or the active ingredient is the antigen that first of all needs to be identified. And there are various ways where it can be identified. So, and when it is identified, either through recombinant technology, that is, it is produced by recombinant technology in the lab to look like exactly like a pathogen. A pathogen can be a virus, can be a bacteria, or so on and so forth. So they will develop something like that, that if this pathogen is injected into the body, it will now elicit a kind of immunity. And that immunity is called active immunity. We know we have two types. We have passive immunity and we have active immunity. The passive immunity is the one that happens immediately, you understand, and take care of the body. It's like a body defense system. If we happen to be exposed as a result of our human interaction and we inject any pathogens, any disease organisms into our body, God has so made it that our body immune responses can take care of it. And how do they take care of it? There are different ways. They will develop antibodies. These antibodies can be of two types. It can be B antibody or T cell antibodies. So, and these B and T cell, they have various other types. The T cell have the T helper cells and the T killer cells. So T4 or T8. I don't want to bore you too much with so many of these details. But after identifying the antigen that can look like the pathogen, we now make that antigen into what can be a sort of use. You put it in a form that it can be injected. So it can be in form of injection. Now they are developing vaccine that can be inhaled, that they just put it in the nostril and you can inhale it, and that's vaccination. It's been done in Africa and here in the UK. Most of the vaccines like HPV vaccine, polio vaccine, flu vaccine is inhaled. You don't have to get injection. So they put it in that form. And they don't give it to human beings first. They have to do what is called preclinical studies or animal studies. And preclinical study, they will get animal that look like human being. They can use guinea pig like rats, or they can use monkeys. That's what I call macaque monkeys. Or they can use non-human primates like a baboon or chimpanzee. So they will bring them in, and under animal rights rule, they will follow all the ethical procedure. They cannot just do anything to animal by cruelty. They will follow all the procedure. They will submit the a protocol for the study to ethical committee, and the ethics committee will grant them the approval to go ahead with the animal study. And they will not give the vaccine to the animal. They can divide the animal into two. Maybe there are 4 animals they want to use, or 8 animals. They will get, okay, this animal, 4 of them, we are going to give them vaccine that we want to test. And 4 of the other animal, we will not give them that same vaccine. We give them another vaccine. Maybe we can give them rabies vaccine. We can give them something else, just to compare. So these two group of animals, we collect their blood sample before we give them the vaccine, and we collect their blood sample after giving them the vaccine at some specific time points. Maybe we collect the vaccine, collect the blood sample maybe a week before the giving them vaccine, and we give them the vaccine today, and we collect their blood sample 7 days after giving them the vaccine, 14 days, 21 days, and 28 days. So after collecting the blood sample, we will now look at the blood sample and see whether the monkeys or the rat develop antibodies against the vaccine that we gave them. You remember I said the immunogen looks like a pathogen that can cause the body to produce antibodies. So they will now measure the quantity, whether one, the vaccine elicits immune responses, produce antibodies, and if it does, what's the quantity? Then the third one is that what is the durability? How long is the immune responses stay? That is one, we check whether it produces. Two, whether the quantity is of adequate measure. And three, the durability. So, and they also check another thing called toxicology. That is whether the vaccine that they are going to give will achieve the minimal inhibitory concentration in the animal, the vaccine that they are giving. That is, will it produce a lethal dose? That is, you identify the antigen, you don't know the dose that will be good enough. You will now give it to those animals. And if you are able to establish that when this vaccine has been given to the animal did not die suddenly, that is, from the lethal dose that was given, that the vaccine achieved the minimal inhibitory concentration, and also that it is safe. So if you are able to answer those three things, then you have successfully completed the preclinical or animal testing. You can now move it to the animal to human testing or clinical trial. So clinical trial is usually preceded by the preclinical or animal studies. So after the animal study, you now move to the human study called clinical trials. And these are also divided into like 4 phases. There's phase 1, phase 2, phase 3, and phase 4. Phase 1 is usually called early phase. It's further divided into 2, phase 1A and phase 1B. Phase 1A is usually conducted in the country where the vaccine is manufactured. It's usually because of a logistic and trust. That is, for example, if you develop vaccine in the UK here and you are now taking it to Africa to go and test, without testing it first in UK, people will start doubting. There's a lot of movement now called anti-vax. You'll be saying that if you get vaccine, it's meant to kill people, it's meant to sterilize the female population, and so on and so forth. That is why phase 1A is usually done in the country where the vaccine is manufactured. So after it is done, in a small number of people, maybe 10 to 20, can go to 25 or 30 at most. You give limited number of people to check the safety in human being. So after establishing the safety, you can now move to phase 2. If it is phase 1 that is done in the country where the vaccine is manufactured, then you move to phase 1B. You can now go and do it in endemic country. For example, if it's malaria, after doing it in country where it's manufactured, maybe in the UK, in Oxford, then you go to Africa where they have vaccine, they have malaria, endemic country, you do phase 1B. That phase 1B, you still do it in a limited number of people. As you saw, I had demonstrated that we did phase 1. That phase 1 is phase 1B. After establishing the safety in phase 1B in limited number of people, you move to phase 2 phase 2 trial. Phase 2 clinical trial, you increase the number of the research participant or the sample size. You can increase it from 100 to like 200 because you cannot make any meaningful finding or conclusion from giving the vaccine to a limited number of people because the genetic composition and other factors varies for human being to human being. So that's why in phase 2, you increase the number from 20 that you did in phase 1 to like up to 200. So after the vaccine has demonstrated that it is safe and it can elicit immune responses in phase 1, phase 2, you move to what is called phase 3. And phase 3 is usually focused on efficacy. Efficacy is about, does the vaccine works? If they say the vaccine will prevent acute malaria, does it prevent acute symptomatic malaria? If they say the vaccine will prevent severe malaria, we want to demonstrate that it is efficacious against severe malaria. If they say the vaccine will prevent against death, we will demonstrate that how many people who receive this vaccine over a period of time that we follow them up develop any of these, either acute malaria, severe malaria, or die. You understand? That is what phase 3 is all about. Phase 3 studies are usually done in more than one countries, and the sample size can be up to 1,000, 10,000 or more. For malaria vaccine, for example, RTSS, it was done in 7 countries and 11 sites, and the sample size was more than 5,000 people or 5,000 children. So then the phase 4, after the phase 3 is done, and you can establish the safety that demonstrated in phase 1, in phase 2, and phase 3, they are still there, and the vaccine is demonstrated to be efficacious. That is, for malaria, there's this malaria roadmap technology that says that a malaria vaccine must have vaccine efficacy of 75% or more to be considered to be an efficacious or effective vaccine. So, and the only vaccine so far that has demonstrated that is R21 matrix M. It has demonstrated that, and when these vaccines now demonstrate that efficacy, it does not end there. It has now to move to the stage of approval. Approval has to be given by European Medicine Agency, and approval has to be given by WHO. They will submit all the safety reports. They will submit all the immunogenicity reports. I happen to work as a member of independent adjudication panel, where we look at all the safety reports on R21 vaccine across 4 countries of Burkina Faso, Kenya, Tanzania, and one other country. So we look at all the children and how many of them develop any safety problem, any severe anemia, severe malaria, any death. Was it due to the vaccine? So, and we submitted that report to the WHO, and WHO now look at it critically, and on the basis of that, they will now give the approval. So when people say that they develop vaccine, they put something there to make people die, it's not actually like that. What I've just described to you from phase 1, or even from animal testing, preclinical testing, to human clinical trial of phase 1, phase 2, phase 3, or phase 4, takes at least 10 years. And it involves different people, different group of experts, different group of scientists. And there's also high level of impartiality, high level of independence, and high level of transparency. All of them will look at it with magnifying lens to see that this vaccine is actually safe for human consumption. So those are the step-by-step approaches to vaccine development. I don't know whether I've answered your question.
Yes, sir. It's been answered.
Okay.
Well-detailed explanation, sir. So I would like that to move to the next question, which is on the slide. The materials will be made available to all the registered participants. We shouldn't worry about that. Then there's a question again that what are the steps required for vaccine development using plant materials?
is the same like as I described. Whether from plants, you need to identify the antigen, and that antigen will be developed further. So maybe if the plant contains one active ingredient, so it has to be studied in the lab extensively to identify that antigen, and that antigen has to be something that can be developed for commercial purpose. Because if you identify an antigen and you cannot produce it for people that more than 100 and more than 1 million people will need it, what's the purpose? So whether it's from plants or is through other means that I mentioned, like DNA or recombinant or RNA technology, is the same step or similar step that is used. The current technology that is used, I saw a question that says that, you see the same level of rigorous development that we undergo for malaria, for Ebola, and for COVID. Yes. For malaria, more than 4 decades, we have been working. Even before I was born, they've been working on malaria vaccine development. I was just privileged to join them, and by the time my career finishes, I was able to see the malaria vaccine becoming a reality. The same for Ebola vaccine trial. But because of the knowledge and experience that we have gathered so much, especially for malaria, so other vaccine technology are now using the same kind of platform that we use for malaria vaccine. So you see, Ebola vaccine that I mentioned to you use similar platform, use adenovirus 26 and immunogen plus MVA as the booster dose or second dose. That's the platform. But there's now a new platform called mRNA vaccine platform or technology, the one that I mentioned that has been developed across 6 countries in Africa. We find out that within a year, we were able to develop, tested, and roll out COVID vaccine compared to malaria vaccine that has taken more than 40 years to be able to establish or develop or roll out. The mRNA vaccine use a different kind of technology in the sense that it is faster. You can fast track everything that I've mentioned, but you still have to go through the animal testing, preclinical studies, and you have to go through the phase 1, phase 2, and phase 3. So the only thing is that the mRNA can easily be done in a fast-track, expedited manner, and that's what happened during COVID-19 vaccine. And scientists are now using that mRNA not only for COVID vaccine. I'm aware of clinical trials ongoing across Africa and in developed countries where they want to use the mRNA vaccine, mRNA technology to develop malaria vaccine. And that malaria mRNA-based malaria vaccine will not only be for children under 5 years of age, but for adolescents and for adults. Because I was made to understand that people are concerned that it's not only children that should have malaria vaccine, but I can tell you that work is ongoing on an mRNA-based malaria vaccine that adults can also use as well as children. Thank you very much. Oh,
thank you very much,
this is
sir.
a very good. There's another question.
Okay, please go ahead, sir.
There's another question. So thank you so much. I'm happy to share a kind of opportunity for medical doctor to build career in vaccinology. There's currently one course called vaccination course in Africa, vaccinology course in Africa, sorry. So It's being run by the group of people that I work with in Oxford Vaccine Group, in conjunction with KEMRI, Kenya Medical Research Institute, and it's taking place in Senegal, Dakar, in Dakar, capital city of Senegal. So it's currently being advertised. I received the adverts about a week ago. I will share it with Dr. Kudrat Adedeji and my brother, Dr. Adegboyega, and they can share with you. Please, I will encourage you to apply. It's a master level vaccinology course, and you are going to be taking through both the theoretical and the practical concepts of vaccine development, right from identification of antigen to the rollout of vaccine, everything that I've described. I have attended that vaccine before in Mali in 2015, and I've been following them up, and I've recommended it to so many colleagues, and they have found it very useful. I want to encourage you to also attend that master level vaccinology course for Africa, and it's being organized by Oxford Vaccine Group, the Jenner Institute, led by Professor Adrian Hill. Sir, please.
Okay, thank you very much. We are delighted. What the differences between vaccine development and drug discovery? I'm just trying to scroll up to the questions that we. Yes.
Thank you very much. So there's a wide difference between vaccine development and drug discovery. So although they follow the same pattern, like I did mention to you, that you have to discover the drug, and in the drug you identify the active ingredient. They call it active ingredient. So in vaccine development, they call it antigen. And you do animal testing, both for drug discovery and vaccine development. And you move to the human testing, the clinical trial, phase 1, phase 2, phase 3, and phase 4. Phase 4 is usually called effectiveness study. That is when you have rolled out either the vaccine or the drug, and you now want to see that the drug is effective. You know, there's difference between efficacy and effectiveness. Efficacy is that you are able to demonstrate that the drug is efficacious in clinical trial control setting. That is, during the clinical trials, you establish efficacy. But in the real world scenario, when you give the vaccine to a child in Mokola or in Ojagbo or in Kigali, will the vaccine still work as if it was in the clinical trials? And that is what is called effectiveness. And that's what phase 4 trial is all about. For example, now the malaria vaccine has been rolled out and is being given all over African countries. Effectiveness studies are also ongoing to see that does this vaccine prove to be effective up to 75%, like the way they've told us about the efficacy. I'm privileged to be one of those effectiveness trial for R21 in Nigeria. You remember that Nigeria adopted R21, and they have rolled it out in 2 states out of 36 states, in Bayelsa State in South-South, and in Kebbi State in Northwest geopolitical zone of Nigeria. Because of a logistic reason, we cannot, and security reason in Kebbi, we cannot combine the trial in Kebbi and in Bayelsa. We focus only in Bayelsa for the effectiveness phase 4 trial in Nigeria. So, answering your question, the steps for drug discovery and vaccine development are similar. But the only difference that I can say is that the length of time from identification of the active ingredient for drug discovery to the rollout of the drug is shorter. It's between 3 to 5 years. But for vaccine, it's between 5 to 10 years. So it takes longer to produce a vaccine compared to drug, but they follow similar steps in their development. I hope I've answered your question.
It has been answered. Furthermore, is the concern about walking someone through the path of vaccinology, irrespective of the background. Is there a pathway to be followed in having this scholarship, sir?
Okay, if you give me some time, maybe I can read it to you. I have and I share it widely with my colleagues because I know Africa, we need more people. So, but what I know that you need is that I think you need to have either first or master degree. Master degree, you need to be working, you need to be an African citizen, living and working in African countries. And you need to provide motivation. That is why you wanted to attend and what you anticipate to gain from that vaccinology course. And third, you are going to have a kind of a supporting letter from either your line manager or your head of department. I think there are two referees. So, by the detail, I'm going to share you with Alhaja Kudirat Adedeji. So, as soon as we finish this, and we will be able to sort of share it with you widely. I encourage you to apply. I think it's still on. It's not yet closed, and it's an opportunity for you to attend that course. And I want to add that that course is free of charge. So they are going to provide you free flights, free accommodation, and the course is going to be facilitated by experts, global leaders in vaccine and vaccinology across the world. And you are going to hear from people who are working in the field, in clinical trials, and also in the industry. They also bring people from Pfizer, from GSK, from AstraZeneca to come and talk. I've been there. They also bring policymakers, bring researchers, and it's a very, very good stepping stone to starting a career in vaccinology.
Thank you very much, sir. Before we move to the next item, I would like to also announce that in addition to the Pasteur Center in Senegal, the EAC Regional Center of Excellence for Vaccines, Immunization and Health Supply Chain Management at the University of Rwanda is also starting the first cohort of vaccinology, MSc September this year. So once we check the University of Rwanda website, all the details are there. FIRAT is also a very strong member of the East African Regional Center of Excellence for Vaccines, Immunization and Health Supply Chain Management, particularly in the development of the curriculum that heralded the approval of the MSc program and other related postgraduate program at the institution. And while we have exhausted what we have on the chat box, this is personal now. So in addition to the story that you have told us, so how do you do your day-to-day management? How do you manage family, relaxation, stress, travels, airport delays, and this and the like, sir?
Thank you very much. Thank you so much for that practical questions. And I want to be honest with you. It's challenging to maintain balance across this, but I always have it in mind that it is important to maintain work-life balance, and ability to do that will demonstrate that you are a true scientist. One should not suffer, and good time management helped me to achieve that. So what do I mean by good time management? I divide my time into 4 priorities. 1, what is urgent and what is important. That one become my first priority. Second, what is urgent but is not important. Then, what is important but not urgent. And third, what is not important, what is not urgent. I remove that from my to-do list. So every day I carry a kind of a small note whereby I organize my schedule. And on the basis of that schedule, I try to manage, excuse me, I try to manage my schedule in such a way that it will give adequate and ample time for my mental well-being, create time for family, and create time for my work and career development. And it is important because most of the time, what we always do is to allow unimportant and non-urgent issue to take most of our time. So if we can prioritize our time on the basis of those 4 priorities, it will help us to free time. You even be operating as if you have more than 24 hours in a day. Whereas good time management will give you enough time to be able to look at what is urgent and important. That's number 1. What is important but not urgent? Number 3. What is urgent but not important? Like it's urgent. If you don't do it now, a life might be lost. That would be number 2. The one that is not important, the one that is not urgent, I remove it. And I don't allow time wasters and time stealers in my space. If you are not going to add value to me, to my well-being, to my mental sanity, you don't have a space in me. So, and I don't spend time on social media that will not add value to me. This I consider time waster and time stealer. And with that, I've been able to manage effectively and maintain work-life balance. I don't know whether I've answered your question.
You did. In fact, far more than it's expected, sir. And I've also learned the first scheme that you have given us today, which is also noted, and it's going to serve as part of our prioritization of our daily activities, stressing the importance of effective time management activities. So we are very delighted, sir. It's as if we should just continue. But I observe that we have exhausted the schedule by 48 minutes.
Wow.
And
I'm sorry for
yes,
that.
No. In fact, it's just like we should just continue. However, just like I posited the other time that the team would work out a physical approach for this continued mentorship plan, and we hope that very soon the details will be communicated on when we'll be able to have a platform from our end, and likewise the Oxford University as well as the London School of Tropical and Medicine, like a partnership arrangement for us to be able to explore further the human capacity need enhancements of the African region in strengthening capacity. There are several activities going on in the region which needs further coordination to be able to deliver on enhancing public health and having a very stronger economy. We hope that this partnership, the initiation of this discussion will lead to very strong partnership that will serve the entire world and also be able to support us in terms of pandemic preparedness, because we can never be too sure what is going to happen. One can never be too sure. When COVID came, like you were mentioning how COVID struck, it disrupted so many things, and up till now, it's like we are still in the recovery stage. And at any time, anything can still happen that can, we hope and pray that will not happen soon. And so the need for stronger capacity development of the African scientists, and we hope that all our partners will be able to work together with us to drive this home. On a final note is to appreciate you once again on behalf of the entire Install Africa team. We are grateful for the time that you have committed and we hope that we'll be able to learn more in the days ahead. So I would like to announce that the next webinar is scheduled for the 26th of June, 2025. The details will be communicated via a registered email. It will also be shared on the website. However, tomorrow we have a session as part of the second session for the Maternal and Child Health Expert Dialogue Series. The second edition is coming up tomorrow, 9 a.m. West African time and 10 a.m. Central and East African time. The details will also be shared via the firat.rw/MNCH. So that's our announcement. We are grateful and delighted that we have come this far, and we hope that we'll meet you very soon. For physical participation to continue the continuation of our series and education. So on behalf of the team, it's my pleasure to thank you once again and then declare this session closed. Bye-bye, sir.
Thank you very much, too. I wish you the best and many more strengths and more successes in all your endeavor. I wish you Godspeed. Thank you very much.
You're welcome, sir. Bye-bye, sir.
Bye.
Thank you. Before you close the session, hello.
Hello.
I would appreciate our speaker for this insightful presentation. Thank you so much. We appreciate for you for responding to our call. I would also like to use this opportunity to tell us the presence of one of the FIRAT board members in person of Dr. Wasim Sajjad, who is a Fulbright scholar at Wisconsin Institute for Discovery, University of Wisconsin-Madison in US. He is an AMR antimicrobial resistance expert, and he has also given us a lecture on this forum. We appreciate his presence. Thank you for also being part of this team. Thank you very much. Our audience, we also thank you for being there for us, because if you are not there, we will not be able to have a successful webinar. We will not be able to have whom to discuss with. We all appreciate you all. Thank you. Once again, I thank Dr. Afolabi for this insightful, beautiful presentation. Thank you so much.
Thank you very much, ma. God bless you all.
Bye.
Bye. Assalamu alaikum.
Bye.
Bye.
Thank you.
Bye.
Bye. Bye. Thank you.